Really excited to get started. We've got a really great group today. So welcome, thank you for coming to our annual JPM Biotech event focused on mechanisms of aging and what those can teach us about different diseases. And this year we're focused on metabolic diseases, on aging as a cause, so obesity, but also related diseases like diabetes, cardiometabolic disease. And this is an area that we at BioAge care very much about. As I'm sure many of you are aware, we have an apelin program going into phase 2, where it has the potential to be combined with an incretin drug to amplify weight loss and improve the quality of weight loss. There's lots of other very exciting mechanisms in this space here, and several of those companies are here today. So really pleased to introduce our panelists. I'll let them introduce themselves, and maybe if everybody could start off with, your name, your title, and a bit about how your work, your current work, is connected to metabolism and to aging. Yeah, Tracey?
Okay, great. Is this on? Can you hear me? Okay. Tracey McLaughlin. I'm a professor of medicine at Stanford, and I'm an endocrinologist, and I am a clinical investigator. So I study insulin resistance, obesity, and metabolism. I trained with Gerald Reaven, who's pretty famous. He's the father of insulin resistance. And I've been there for a long time now, 25, going on 30 years. Yeah.
Excellent. Hi, everybody. Pleasure to be here. Thanks for the invitation, Kristen. My name is Rob Myers. I'm Chief Medical Officer of a Bay Area biotech company called OrsoBio. I've been there for about 2 years, and we are developing several oral compounds for metabolic disorders, including obesity, diabetes, dyslipidemia, and look forward to telling you more about those programs in a second.
Thank you, Kristen. Thank you for the invitation. My name is Michael Davidson. I'm a cardiologist, lipidologist. I'm also the CEO of NewAmsterdam Pharma. We're a Dutch NASDAQ-listed company that's developing a CETP inhibitor. It's a cholesterol ester transfer protein inhibitor that lowers LDL cholesterol and dramatically raises HDL cholesterol and lowers LP(a). It's also a longevity gene, and I also see patients. In fact, I run the lipid clinic at University of Chicago. I'm a professor there, and I'm flying on the red eye tonight to be back for clinic tomorrow.
Yeah, my name is Axel Haupt. I'm an endocrinologist. I'm Senior Vice President at Eli Lilly and Company. I'm heading early phase clinical research, and we have an exciting portfolio in ASCVD with sRNAs in LP(a) and other lipids. Of course, we have a thriving incretin portfolio with retatrutide, orforglipron, and we are in cardiac heart failure. So a lot going on in early phase in Lilly.
Thanks, everybody. So we're going to start with a bit of a stage-setting question. So when you think about aging as a risk factor for metabolic disease and the interplay from a biology perspective about the pathways that are important in obesity, important in metabolic disorders, and how they change with age and how they drive aging. And Tracey, do you want to kick us off here?
So the pathways, you mean, so maybe draw some commonalities between metabolic disease, obesity, and aging?
That's right.
Yeah, it's really interesting. Well, I mean, there are many ways you can look at it, but first of all, the conditions that are comorbidities of obesity are all also diseases of aging, right? So diabetes, hypertension, cardiovascular disease, cancer. People forget that cancer is very highly related to obesity. In fact, with bariatric surgery, the decrease in mortality is primarily due to decreases in cancer. Is that interesting? I mean, there's cardiovascular disease and everything, but NAFLD or NASH, they're all diseases of aging. And if you get down into the molecular pathways, there are actually a lot of the same molecules. Like if you look at the fat and some of the pathways that are activated. So for example, inflammation, which is thought to play a role in aging, all parts of aging, all organs, there's inflammation in the fat, and there are circulating inflammatory markers. And obesity is often said to be a disease of, or a condition of low-grade chronic inflammation, or immune responses. So people who are obese don't respond well to vaccines. They have an impaired, B cell and T cell response to vaccines. So there are a number of parallels from molecular pathways to immune responses to the presence of the same kind of diseases of aging.
And Axel, I know you had some thoughts on inflammation here as well in this context.
Yeah, you know, inflammation is, I think, one hallmark of aging, and I think controlling it with inflammation targets or obesity treatments is, I think, an opportunity which now arises with the new incretins. But I'm also very excited about Lp(a), so there is also a connection to aging, and let's see if there the new drugs can have a dent in that as well.
Yeah, and Michael, you mentioned briefly that CETP is an aging target. It's enriched in centenarians, certain variants. What do you think about its general role in aging?
So we're excited about CETP from a aging perspective. It was first noted in the Bronx aging study. In fact, these are several hundred Ashkenazi Jews in New York that all lived to be 100. One of the first women died at 118 this past few months ago who had the CETP loss of function gene. What's important about that is the gene, obviously you have high HDL and you get low LDL, but you also get low, very large LDL particles, which are part of the signs of that. But we also believe that we're working on is that CETP through the HDL raising mechanism will prevent or maybe treat Alzheimer's disease, especially ApoE4 related Alzheimer's disease, because the HDL, we believe, there's no LDL in the brain, it's all ApoE and HDL. And so you need to get HDL to go into the brain and help remove cholesterol. It's not being adequately done by ApoE4, those have ApoE4 gene. So we think this dysfunctional gene can be improved by using a CETP inhibitor. So we have some proof of concept data in humans now that we're quite excited about that. But yeah, it's a very prominent longevity gene. And getting back to inflammation just for a second, my previous company, Corvidia, interleukin-6, it's another longevity gene. So like and IL-6 plays a very important role in aging. As you get older, it goes up. We think that high IL-6 is what causes low LDL. That's linked to higher mortality. People thought for a while that low LDL could be bad because of higher mortality, but it's probably linked to the high inflammatory state that exists in that setting. So Corvidia was bought by Novo, and now developing ziltivekimab for cardiovascular disease and inflammation. So very excited about that.
Yeah, thanks. And we're segueing a bit in the next question now, which is really to talk about some specific drug targets that are aging drug targets, but that are in the clinical pipeline, right? Because there's a few of these that aren't necessarily recognized as such. CETP is one. I would count FGF21 as well, right? Like that's a wonderful NASH target, but also clearly an aging gene. And Rob, I wanted to ask you about mitochondria and protonophores.
Yeah, I didn't mention in my earlier remarks that we have a portfolio of mitochondrial protonophores that are, the focus is really enhancing energy expenditure to treat obesity and obesity-associated disorders. And I think to Tracey's comments earlier about the sort of common mechanisms in obesity and aging, inflammation, mitochondrial dysfunction, enhanced reactive oxygen species are important to both, and we believe that increasing energy expenditure with a mitochondrial uncoupler can address those aspects of the pathophysiology of aging as well as obesity. There are some preclinical data suggesting that uncoupling can prolong lifespan in mice and other species. So I think there's a clear link there between the mechanism and aging.
Cool. And Michael, I wanted to ask you about Clotho as well.
Yes,
Another
right.
one. Yeah.
So we, again, it's all from my clinical experience. We had a very well-known family whose mother just turned 100, and she smoked tobacco cigarettes a day since she was 15 years old. In fact, they think their father died from her secondhand smoke. But she's 100 years old, just had her birthday, and so wanted to look at her genome and find out why despite these very unhealthy lifestyle habits, has a very high cholesterol, by the way, too. So Clotho stood up as one of the genes that she had. So I got very interested in Clotho, and I know we don't understand the mechanism, but it's certainly clearly another gene that's been linked to longevity and reduction of Alzheimer's and ApoE4. So we licensed it, and now we're moving it forward into preclinical studies and trying to understand the mechanism and the dosing, and we hope to get into human trials in about a year and a half. That's where we are with the clotho protein.
Very exciting. Yeah. Any other thoughts? Yeah.
Yeah, I can also add some targets, right? I think functional muscle mass is really important to mention for aging, and preserving it or building it is really key, I think, in this aging process. And that is also closely linked to quality of life and functional benefits. And obviously, apelin is a very interesting mechanism here, and we are also very interested, for example, in ACT2/3, and we in-licensed bimagrumab, and both mechanisms can be really ideal partners for our incretin franchise, because rapid weight loss is obviously associated with some lean body mass loss, and preserving during that body mass loss, that lean body mass will really prevent, I think, or will help patients to be more functional and maybe age less, right?
Yeah, definitely. I mean, the profile of weight loss can be very different depending on age, right? I think when older people lose weight for pretty much any reason, they lose proportionally more muscle, and that which can drive poor outcomes like frailty, like loss of independence. Yeah. Let's move on specifically to incretins. They've had a really good year this year, and they've had huge impacts in diabetes and obesity from a treatment perspective. But what about the future, and what about their ability to prevent certain diseases like cardiovascular disease? There's been some really exciting data readouts in the last year, and also some news articles with spicy titles like, are incretins our best longevity drugs? Because in a sense, right, they have the potential to shift the health of populations. What do you guys think about that? Yeah.
Well, I can comment since you're biased. I'll give you my objective, unbiased view. I'm really excited about these drugs. I mean, I truly believe that they could change the global patterns. Diabetes has gone up and obesity has gone up for decades. And they could change the tide. We might see diabetes actually start to go down. All drugs that cause weight loss, for the most part, do prevent diabetes. I mean, when they've looked at liraglutide, I think semaglutide was also shown in a secondary analysis to prevent diabetes. And, it's just weight dependent, right? So if you lose weight, the studies have shown with metformin and dietary weight loss and numerous drugs that you can prevent diabetes. So that's a very, very important change in the tide, and also a very cost-saving change. And that's not the only one. So I think the place where we are, and the muscle mass is really important, and the frailty, because as a clinician, you see that when people age, if I have an 80-year-old, I don't really care if their glucose is a little high, but I really care if they fall, because then they get pneumonia, break their hip, and they can die. So that is really important just clinically. But I think where we're headed with the drugs, and something to think about is, do we want 2/3 of our population to be on these drugs, and do we want them to be on these drugs forever because they're going to regain their weight when they stop the drug? I mean, that's going to be confronting us in the next decade, and we may need to figure out ways to enable people to maintain their weight loss, or to stratify who is going to benefit most from those drugs, right? We have our healthy obese and our less healthy obese, and we have very unhealthy overweight people. We have normal weight diabetics. So, I mean, we're kind of have to move into an era where we figure out how to work with this large class of drugs. Now you probably have some comments.
I think you're spot on. I think we have, solved, or retatrutide may solve for most patients the weight loss to achieve, but the maintenance is I think, an issue, and obviously you need to stay on those drugs to maintain, because we are not changing the set point. So we need additional mechanisms and partners to avoid metabolic adaptation, to increase energy expenditure, to really not have the appetite drive after stopping incretins, right?
Yeah, and I kind of think, I don't know what you guys think, but that point where your body's adapting, so your body's adapting, right? So you're losing weight, you're in a calorie deficit for all this long period of time, and then you stop. So it may be that even during the first 3 months, which is when that trajectory of weight regain is high, that might be a really critical time to intervene with, targeting some other pathways. I don't know if you have thought about that. I mean, there are many. Or it may be more long-term, but I think this is what we're looking at now. We don't have the answers. We don't even know exactly why people are regaining their weight.
I think it was very exciting, the SELECT trial for the 20% event reduction. No LDL lowering, so I think people have this misunderstanding that you lower weight, you lower LDL, that they're not connected mostly. And so in my lab near Chicago, we actually looked at other mechanisms where GLP-1 can affect atherosclerosis. GLP-1 receptors are on the macrophages, and GLP-1 agonists do in fact increase efflux of cholesterol out of macrophages. So I think there are many mechanisms going on why the benefit was so great and early with the SELECT trial. It's not just the weight loss, it's the anti-inflammatory effects. I think it's direct anti-atherosclerotic effects as well through these other mechanisms that make this class really exciting. And as a cardiologist, I can say, I hope that my fellow cardiologists have always been reluctant to jump on the weight loss, even diabetes drugs. And I think now with data like this, you'll see more and more specialists like cardiologists are going to become more evidence-based and use these therapies much more readily in practice.
Yeah, and we need to show the data that we really impact heart outcomes, right? So there's a lot of studies going on, and I think semaglutide has shown ASCVD benefits in obese population, has in HFpEF shown nice outcome data. We will disclose Summit this year. This is another HFpEF trial. We have, I think, also in ASCVD trials ongoing. We have shown promising data in kidney and URCR reduction and slowing down slope of eGFRs. So I think there's a lot of promise, but we still are in the phase of accruing data for heart outcomes for the incretin space, right? And I think it's a lot of promise.
We're watching it all unfold right now, right? And for those of us in the aging biology space, what excites me, what I think excites a lot of other founders is the prospect of discovering mechanisms that can be used to ultimately prevent disease, or sort of intervene earlier in the process. And we used to only really have the example of a statin. We can point at statins as a therapy that was ultimately, initially treating a disease, now preventing disease. And we have one more example. And just sort of thinking more broadly, what are the barriers to studying new drugs in a preventative paradigm as preventative medicines? I know that, Michael, you've thought about a diabetes prevention trial. I would love to hear some thoughts on that.
Right. I think it's going to require, maybe cardiologists are the victim of our own success because we want to see evidence of benefit that are substantial, outcome benefit, MACE, major adverse cardiac event reduction. And so that's a really tough hurdle, and it takes a lot of money, capital, time to prove these events. So unfortunately, I've been small biotech, this is my fourth company, small biotech. It's really hard for small biotech to have that type of capital resources to do these type of studies. So we need big pharma to be there, and we can't always guarantee that's going to happen. So it's really, look at all the different companies out there in the biotech world. I don't know how many are cancer, how many are gene therapy, and very few are cardiometabolic, and that's because of this hurdle that we face of financing these large trials. So we have to, I think, come together, regulators, pharma, biotech, academia, to really figure out a way to find other ways to answer these questions in a much more cost-efficient way.
And even if you do answer them, right, you get over that regulatory hurdle, you run your large outcomes trial, then what about reimbursement? Like, how do we think about that?
Yeah, I think you're right. So first of all, to study prevention, you're studying a non-diseased state, which maybe the people who are investing in your company or the people backing it or even the NIH, people aren't as interested in that. It's not reimbursable to say you have metabolic syndrome or even, I don't even think obesity is unless you have multiple other risk factors that you can pinpoint. So we sort of need a shift in mindset, but that's a problem. And the other is just the duration. So all these trials, including the GLP-1 trials, most of the drugs, any cardiovascular drug, are being done in people who either have established cardiovascular disease or 2 risk factors generally, because you can't wait 20 years for an event. It's just too expensive. Or you do a shorter trial, you need 1 million people because the event rate's so low. So those are really, so to find other ways, at least to do the first trial, is a really good idea. But then you still need a randomized controlled trial to get approved by the FDA. So I think it's really tricky to get into prevention.
No, I agree. So this is really an issue, right? And if we want to go to premodal prevention, right, with gene therapies like WERV, where you added PCSK9, how to show the ASCVD benefit 40 years later if you have one stop, right? So we need to solve those problems, how to do it. But obviously, even primary prevention trials, as you said, are high-risk trials to ensure a high number needed to treat, that payers will pay for the therapy, right? And that the trial size is not, too large, right? Because if you don't have an event rate, you don't show a benefit. So that there needs to be a balance. And obviously there need to be also movement from regulators and the FDA to give us development paths and biomarkers, which then you could suggest there's benefit, because we may not be able to provide the hard outcome for those patient populations.
The only thing I would add is that from a patient perspective, to be on a therapy for prevention for a disease which is maybe asymptomatic is sometimes a tough sell. Michael, you could talk to this regarding putting patients on statins, et cetera, for primary prevention of cardiovascular disease. So it's imperative that you have a well-tolerated therapy, probably an oral therapy. I don't think, despite the select trials showing SEMA preventing new onset diabetes, but reducing it by roughly 75%, to convince asymptomatic patients to go on a GLP-1 for that indication is going to be tough, at least injectable. So.
They will do it because of weight loss and body shape, right? Not for the prevention of diabetes.
Yeah.
So that's the beauty of incretins. I think the compliance is much better for this class.
Even the incretins is not that great. At 1 year, it's about half, right? So, but that's because of the side effects. But if it weren't making them lose weight, probably no one would take it, right? Who wants to take a shot?
Right.
Like if you compare it to insulin, no one's signing up for insulin. But I mean, so to do something for your health that you can't feel at an early stage is hard.
Yeah, this is a great transition to the next topic, right? Because these are huge trials, and these are huge indications, right? And trials that take a very long time. So let's talk a bit about biomarkers, right? Like what's sort of the state of the art there? What can we expect over the next few years, both in terms of biomarkers that enhance clinical decision making, as well as things that are potentially surrogate endpoints?
Okay, well, I'm going to get on my soapbox. We are spending a lot of money and effort, including my own lab with Mike Snyder, to try to find fancy biomarkers or panels, a targeted omics panel, which could be great, right? You have these kind of things for early detection of cancer. But for metabolic disease, you can actually do it pretty easily with triglycerides and HDL. Any of you cardiologists who work with Ron Krauss know this, but a high triglyceride and a low HDL reflect a metabolically disordered state, including high ApoB, small dense LDL, and insulin resistance, which goes along with everything. And so, or you can measure an ApoB. I mean, you can measure those two things and I think have a pretty good idea of what's going on. Glucose is not good. Insulin would be potentially good, but it's not standardized across labs. So I think there's some very simple things, including, you could try to do some anthropometric things. Body weight's not perfect. They tried to do this with that ATP3 criteria and metabolic syndrome, where you're kind of putting 3 out of 5 things together, and if you have that, you have a high risk. So I think there are simple biomarkers. Of course, we always want to get better ones, and perhaps you guys have other ideas. But I just wanted to put in the plug for we do have some simple biomarkers at our disposal already. We just aren't really using them.
I can add my experience. I didn't mention in my introductory comments, but I'm a hepatologist, and I used to work on NASH at Gilead, leading the liver fibrosis programs. And we banged our head against the wall for a long time trying to develop biomarkers of liver injury. And right now, if you're not aware, the standard for approval of NASH drugs is liver biopsy. And so while we came up with biomarkers which could actually quite accurately predict the stage of disease, the amount of fibrosis in the liver or fat in the liver, the challenge really was convincing, or has been convincing regulators that a change in these biomarkers is relevant. And so to get a biomarker qualified is an extremely challenging process.
Yeah, I mean, I also like what you said about lipids, ApoB, LDL particles. CRP, I think, is a really good, but unfortunately not very specific biomarker. It really correlates very well with mortality, morbidity, mortality. In the cardiovascular world, we've made major advances in CT angioimaging in the past few years. We actually can look at types of plaque, non-calcified plaque, even among the non-calcified plaque, plaque that's more vulnerable for rupture. So I think we just had a FDA feedback on a study we wanted to do with using CT imaging, and the FDA said, not yet. I mean, not yet as a regulatory endpoint, unfortunately. But I do think this is the future, because I mentioned all the big trials we have to do, hundreds, thousands of patients. But if we could just show, plaque regression in an imaging study, that would be a lot quicker and a lot easier, a lot less expensive. And again, if the stakeholders come together and decide if you're seeing the plaque melt away on imaging, that should be adequate to get registration. I think that would be a big step forward. But we're getting there. I think we are going to get there ultimately. As long as we have kind of like LDL being supported as a approvable surrogate, but yet I still think, even though we have 8, 9 different mechanisms of lowering LDL, all showing a benefit, there's still this school of thought that you have to have evidence for every single one of those LDL lowering benefits. And so that's where it gets, I think, a little bit too much. A little bit too much evidence needed to really say LDL lowering, no matter how you do it, should achieve a benefit.
I mean, how long did that take to get LDL accepted as a validated surrogate? I mean, you lived through that.
Right. It's been 30 years, but just the meeting at the FDA just in December, they asked, is LDL now a surrogate where you don't need any outcome studies? And he said, not yet. Not yet, believe it or not.
Payers want to see the outcome as well, despite LDL being a very, very relevant biomarker, right? So I think this is increasing research costs and development times, right? So that needs to be always clear if we don't accelerate those biomarker things. And no one doubt LDL is important, right?
That's weird. I think I see these analogies. So one that's similar to yours is insulin resistance. So where that failed, because there was a drug, rosiglitazone, that was extremely effective at reversing insulin resistance, but it increased cardiovascular mortality. So it's trials like that that make it difficult when you're trying to look at a surrogate marker. But glucose, on the other hand, while a lot of trials at the beginning had to show not only glucose lowering, but a reduction in microvascular disease, so that's organ damage from the high glucose. I don't see anybody requiring that anymore. They just need to see the A1C lowering. So however you get there is okay if you can lower the A1C. So like with diabetes, I feel like we sort of got there,
So
I
think it's a bit more than this. So we have large exposure for a normal phase 2 program needs to have, I think, 500 patients reaching 2 years, and they need to have CKD or high-risk ASCVD. And in those patients, you need to show that you don't increase, basically, events. And, of course, we have a total exposure by launch, which is much larger than several other programs.
So you mean in addition to glucose lowering, you have to show it doesn't increase cardiovascular events?
Yeah.
That's true. That's true.
This is, you need to establish cardiovascular safety, but now it's standard that we go for superiority, and I think that you don't need to do before approval, but it's normally ongoing during phase 3 already. Yeah.
So Axel, like, what is the Lilly take on future biomarkers? Like, is that an area you guys are investing in? Do you think we already have some good ones and you're just implementing those, or are there sort of very active research there as well?
It really depends on the disease state where biomarkers are established. But for example, in HFpEF, you don't really have a good predicting biomarker, and this makes phase 2 really risky and phase 3 even more risky, right? But if you have an established biomarker like high-sensitive CRP or LDL or ApoB for your compound, right? So then you have good de-risking in phase 2, which is obviously nice for downstream investments in phase 3.
Yeah, it's still useful.
Yeah. In the Alzheimer's world, because we're doing this in our trials, and we're very excited about the tau, 217, 181 biomarkers. I think Alzheimer's is a disease where it really is important to prevent it rather than try to treat it. And the treatment we just got approved is marginally beneficial and has a lot of side effects. And so I think that in this situation, I think the regulators are getting very supportive. In other words, if you have positive benefits on these biomarkers, you can maybe get registration. So I think Alzheimer's is moving in a very biomarker-driven way for regulatory approval, which I think is great because I think we know that the impact of that on health and longevity is so enormous that if we could figure out a way to prevent it and using biomarkers to get the early stage of disease and then treat those, that would be a major improvement.
Yeah, earlier trials and faster readouts. Yeah. All right, so we're just going to have one more question before we open it up to audience questions. I'll keep it short and sweet. So just to end here, this is a very new, very quickly evolving space. There's lots of novel mechanisms in the clinic, and success of the incretins have really changed the conversation. So what are you looking forward to, say, in the next year? Like, what kind of data readout is going to happen that you think could be a really big and meaningful for the field and change the way we think about this space? Yeah, who wants to go first?
I mean, something that Axel alluded to earlier on is their study of bimagrumab plus an incretin, this combination effect. Can preserving muscle or building muscle enhance the effects of a GLP-1 receptor agonist? I think that's a super important study for me because I think, to my knowledge, this will be the first combination study for the treatment, recent combination study for the treatment of obesity. And we're very much focused on developing new mechanisms of action to combine with GLP-1s. And so I think that's going to be very informative of a first step for how does the future look?
For me, it's the Horizon trial. Well, it's 25, early 25. It's the Lp(a) lowering trial with the ASO. Again, Lp(a) meets all the criteria of validated genomics and causal relationship. And now we have a first intervention trial with an ASO, and outcomes will be read out, and that'll then move Lp(a) to the modifiable risk factor status, which I think will open up another residual risk reduction that could be quite significant for patients.
I think I'm very excited about our Lp(a) phase 2 data. So we have McLaughlin and our siRNA reading out this year. So really important readouts. I'm very intrigued by our BELIEF study. So this is the study where we combine BIMA and semaglutide. We'll read out, and obviously we have also other important pipeline progresses in phase 2. So a lot of exciting data coming out of the Lilly pipeline. But I'm also hoping that we may have an outcome readout for tirzepatide in the next coming years. Yeah.
CV outcome trial.
So we, as you know, we have in type 2 diabetes and kicked off an outcome trial. And that's the first trial where we compare it against an active comparator that's dulaglutide. So it's a high bar. And we have here a superiority trial powered. So I'm excited about that trial. I don't know, it's an event-driven trial. You never know when they read out, but that's exciting.
I think what I'm really excited about, and this will take more than a year, but it's really having a more precision medicine approach or precision health approach. Health is just lifestyle in more of the preventive stage. Partly because we have such a large array of treatment mechanisms now that all target different pathways. So for example, in diabetes, we just had insulin and sulfonylureas, and then we got metformin, but now we've got drugs targeting multiple different pathways. And type 2 diabetes is, in the next 10 years, will probably be subclassified. There's a lot of work on that right now, and you can subclassify it different ways, but if you can subclassify it according to the underlying physiology and then attempt targeted treatment, you'll have better adherence, better success, quicker, getting your A1C to target. And the same thing with weight loss. I feel like we are forced to face, like I mentioned earlier, this huge number of people who may benefit from weight loss, but maybe everybody doesn't. I think we really need to get back to this idea of kind of targeting the highest risk overweight and obese, and that BMI alone is not really sufficient to decide who needs to lose more weight or lose weight at all. So I think we will be forced to deal with that and maybe look more to metabolic markers of disease, so who's really the unhealthy person who really needs to lose that weight. Anyway, so I think we'll see that. That's also going to lead, to targeting, right? Targeted weight loss, targeted lifestyle, targeted drugs.
Yeah, thanks so much. Let's thank our panelists.